for aquaculture. Dr. Nandini will talk about it, because how many individuals can be produced in 1 milliliter, and what are the favorable conditions. She will talk about them. Then when the conditions are not favorable, they produce another kind of female called amictic female. However, it produces the haploid eggs. These haploid eggs, if they hatch, they become males. If they are fertilized, they become the resting eggs. These resting eggs are the same as the artemia. The cell division has already occurred in this, but organogenesis has not started. So in this case, they can undergo dormancy for a very long period of time. Records say we have about 40 to 50 years time. In case of artemiasis, you have up to 1,000 years they can survive. Although hatchability decreases with age of the resting egg. And the resting eggs invariably hatch into females, and then the cycle starts. The whole process of hatching resting eggs in case of rotifers is different from that of artemia, because resting eggs faithfully do not hatch. We have about 60% to 70% success, whereas in artemia you have 100%. So if you buy 1 kilogram of resting eggs, only 600 grams of neonates are born. But still, investigation is going on how to hatch 100%. There is some, at least in one species, Brachyonus calciflorus, the achievement is nearly 80%. Now, what type of research can be done on rotifer? There are two. For any organisms, possibly is true. So some aspects are basic, some aspects are applied. In this case, we will talk about applied aspect of ecotoxicology and aquaculture, or at least mass production of zooplankton. Some part Dr. Nandini will talk about it. And basic aspects of species diversity. How many species of rotifers we have in India? Only, for example, maybe 400. How many species we have in the United States? So this is a prestige, as one of the speakers already mentioned. I think Dr. Kavita already mentioned, it's a question of biodiversity. And so we need to know each group, how many species we already have in each country before they disappear due to inhuman activity by humans, especially the contamination. Okay. Then they are also evolutionary biology, they are important. And then water quality indicators, this talk I will give separately towards the end. And then some of the basic questions students usually ask, and some of the interested persons also ask, what is the smallest size of rotifer? Approximately the neonate will be something like 30 micrometer. And each rotifer has approximately 1,000 cells. And you cannot count the cells because it's a syncytial system. You can only count the number of nuclei, they strain them, and then you can count them. And the rotifers do not swim very fast. They are weak swimmers. At least they swim at the speed of 1 millimeter per second. Therefore, weak fish larvae can easily catch them. Fish larvae swim about larval fish. Most species swim about 10 millimeter per second, whereas the rotifers 1/10 of that one. Therefore, swim larvae in the young stages also can catch the rotifers as a prey. And the rotifers, surprisingly, rotifers do not sleep, not even 1 minute. If you find a rotifer sleeping, it means it's dead. From the birth to death, they keep on filtering, doing what is ecosystem function in aquatic ecosystems. That is transferring energy from the phytoplankton to the fish larvae. On an average, in a given water body, you find approximately 50 rotifer species. Much depends on your ability to taxonomic skills, but on average, about 40 to 50 species can be easily found unless the water body is highly contaminated. Each rotifer species can, each individual of a rotifer can consume approximately 2,000 cells per hour of the green alga. And the rotifers normally live between 30 to 40 days. Most of them die before 25 days. Then the cyst can survive up to 40 to 50 years, and then each rotifer can produce between 40 to 50 eggs in its lifespan. And now high density cultures, Dr. Nandini will talk about it, and in 1 milliliter, you can produce approximately 25,000 rotifers. This number is enormous, and in high density cultures,